Guys, welcome to our channel. This is Science Lectures. Please, thanks for being with us. Consider subscribing and like our videos and comment in the comment section. Please consider following us on our social media handles. Twitter, you can follow us on Twitter. Thank you, please, for joining us. So we are still continuing with our Monera, Kingdom Monera, and we are still dwelling into the concepts of what we refer to as bacteria. So we want to have a deeper understanding of what bacteria is. But today we have a special type of bacteria we are looking at, and it is called Archae bacteria. And we want to know what this Archae bacteria really is. So Archae bacteria is a form of bacteria that Archae bacteria, form of bacteria that will survive under in extremes of conditions. So the history of the Archae bacteria is a certain scientist by the name of Woos, and this scientist named Woos discovered and invented the division of the Archae bacteria as the third kingdom of classifying microorganisms. Remember the kingdoms: Eukarya, then Prokarya, then Archae. So he discovered it as the third one, which is Archae. So who's in around 1977, discovered that there are certain bacteria that can survive in extremes. So this guy was in Alabama, Illinois. So extremes of conditions, we are going to look at such bacteria that have the capacity to survive in the extremes. So we'll start with the special. So we look at these unique types of bacteria. We have the ones we call thermophiles. Thermophiles, what are thermophiles? Thermophiles are bacteria that will survive in extremes of temperatures, very high temperatures, the ones that will survive in high temperatures. For example, temperatures above 70 degrees Celsius. So such bacteria are found in hot springs, and in areas where we have volcanic eruptions, So scientists wanted to research whether there are certain bacteria that can survive in such conditions. However, what fascinated them is that they found out that some bacteria can really survive such conditions. So thermophiles, for example, we have bacteria known as Thermus aquaticus. So Thermus aquaticus is the prominent one, well known, that survives in extremes of high temperatures. So this Thermus aquaticus, however, is very important because we can use it in the formation of what we call DNA polymerase or tag polymerase that is used in the PCR reactions, polymerase chain reaction, to yield multiple copies of DNA that maybe we are studying about in a scientific experiment. So this is a thermophile. However, another area where we can find these thermophiles is in the vents down in the ocean, the deepest layers of the ocean, where we have eruptions taking place in those hydrothermal vents. They are called hydrothermal vents. We can find such bacteria, the halophiles. So halophiles, extremes of temperature. Then we also have other bacteria. However, bacteria that survive in very low temperatures, before we go further, in temperatures of around very low temperatures, 2 to 3 degrees Celsius, down there, low temperatures. Such bacteria are called psychrophiles. And these psychrophiles, surviving in very low temperatures, we can find them in areas like the Arctic and the Antarctic, in areas that are really stellarized, those regions that are really stellarized. So we can find psychrophiles surviving in those areas of very low temperatures. So that is the first, what we can have a look at on the thermophiles. Then we also have other types of bacteria that are under Archae, the acidophiles. The acidophiles survive under regions of very low pH, pH even below 2, a value of pH below 2. So acidophiles, very good at surviving in regions with very low temperatures. These are areas where for them they will thrive. Then we also have at the opposite of acidophiles, a bacteria known as alkalophiles, the ones that will survive in high pH, a higher pH of about 10 to 11, a value of 10 to 11 pH. Then we have other bacteria called halophiles. Halophiles is another division. Halophiles are the ones that survive, bacteria that survive or thrive in areas or regions of high salt concentration. So where we find high sodium chloride concentration, we'll find halophiles surviving. For example, cyanobacteria. Cyanobacteria can survive in high salt concentrations, high sodium chloride concentration regions. Then under also the Archae, we still have another type of bacteria, and those are the methanogens. For examples we have methanococcus and methanospirillum. So these ones are methanogens, can survive. So these methanogens, for them, they are anaerobic. In fact, they are obligate anaerobes. Obligate anaerobes. They are obligate anaerobes. That means they entirely require no oxygen to do their process of breaking down the organic matter to methane, or what we call biogas or natural gas. To methane or biogas or natural gas. So these guys are very important because they can be studied. They are studied to see if they can do bioremediation, good bioremediation. However, they can also be used in sewage treatment, can also be applied in sewage treatment. So that is it for Archaebacteria, a specific type of bacteria that somehow differs from these other bacteria. But what causes the difference between Archae and these bacteria is in their genome and their ability to survive in extremes of conditions. We are going to look at these bacteria survive too much. So we are going to look at the success of bacteria, the reason for the success. Why are they so successful in whatever they do? So success of bacteria is what is our factor now disturbing. So we want to solve why bacteria are successful. We need to know. Number one thing why bacteria are successful is because of their reproduction. In our video of reproduction, we talked about the fact, if you've not watched, please you can go and watch reproduction in bacteria, sexual and asexual. Reproduction, we say, bacteria can reproduce by what we call binary fission. And in binary fission, we can have multiple bacteria being produced. So we can have multiple bacteria being produced, a number of them. So the reproduction, it being binary fission and very rapid, it brings about, it causes multiple divisions. The rapid divisions will bring about the success. Because if this bacteria can reproduce rapidly and divide rapidly, let's say we are having one bacteria developing into two, then we have 16 bacteria. So this division is too rapid. That makes them successful in whatever environment they are. But however, for this to happen, we say all nutrients should be available. the nutrients should be available, and no factors limiting their growth should be present. Which other thing makes the bacteria too successful? When conditions are harsh, bacteria form what we call endospores. Endospore formation. Endospores are used in surviving harsh conditions. For example, UV radiations that are tough, then limited nutrient availability. Nutrients available, limited UV radiations are present. Presence of toxins can also make them form the endospores. Endospores are resistant to temperature. Resistant to temperatures. They are resistant to temperatures, and they can survive for a long period. A typical example of a bacteria that forms endospores that are very resistant and very dangerous is called Bacillus anthracis, the one that causes anthrax. So formation of the endospores tends to protect the bacteria from the harsh conditions. And then which other thing leads to the success of the bacteria? Some bacteria are capable of producing what we call, all bacteria produce toxins. These toxins will harm the host. However, a specific type of bacteria, that the ones that are gram-negative, produce a more serious toxin that is very dangerous, which is an endotoxin. This endotoxin is produced by specifically gram-negative bacteria, and an endotoxin is deadly as compared to the exotoxin that is produced by the gram-positive. So we have an endotoxin and exotoxin. Endotoxin is very dangerous and can kill the host. For example, an endotoxin produced by this bacteria called Clostridium botulinum, the one that causes food poisoning. A microgram of this bacteria can lead to harming of the host. So production of the toxins has also been shown to provide success to different bacteria. Then which other thing leads to, another thing is mutation, ability to mutate. Mutation, the rapid mutation in every cell division. When the more that the bacteria cell divide, the more their genome mutates. And the more the mutation happens, the more they can adapt to the changing environment. So this one can also be a factor leading to success of bacteria in the environment. Then bacteria have what we call a plasmid. And a plasmid, we describe a plasmid as the extrachromosomal DNA. It is extrachromosomal DNA. And this extrachromosomal DNA is responsible for what we call antibiotic resistance. So due to their capacity to resist the action of the different antibiotics, it makes them survive. Then why again a bacteria capable of surviving is due to the presence of what we call the capsule. The capsule is the outermost covering, and this is the outermost covering of the cell envelope. And this capsule helps in what we call, what is the virulence? It is a virulence factor. That means it prevents attack by the host cells. So the capsule is basically important in preventing phagocytosis by the host cells. And bacteria that produce this capsule are not easily manageable within the environment. That is why there is development of vaccines for some of the bacteria that is, for example, Haemophilus influenzae. For example, Clostridium, meningitis, meningitis, the one that forms, the one that causes meningitis in humans. There is development of a vaccine for it because it can produce the capsule as a virulence factor that prevents phagocytosis within these cells. So a capsule is very important for survival. Then finally, what can also be important for the success of this bacteria? Typically, we look at the gram-negative bacteria. These gram-negative bacteria have a layer of lipopolysaccharide, lipopolysaccharide, and this lipopolysaccharide is capable of producing what we call the beta-lactamase enzymes, which beta-lactamase enzymes affect the action of penicillins. So penicillins might end up not working on the gram-negatives because they have a lipopolysaccharide that aids in producing the beta-lactamase enzymes. That is why in some medications, a combination of a penicillin plus a beta-lactamase inhibitor, like clavulin. So you will see combinations on market like amoxiclav, which is a combination of amoxicillin, the penicillin, and clavulanic acid, which is a beta-lactamase inhibitor, just to effect the working of the drug. So those are some of the reasons of the success of this bacteria. Reproduction is too rapid. Formation of the endospores that are resistant to even high temperatures, to radiations. They can survive for longer periods in the soil. Then toxins, production of toxins, most of the endotoxin that is very dangerous for living organisms. Then mutations. The moment the bacterial cell replicates, it can have random mutations in its DNA. Then the plasmid that is responsible for antibiotic resistance. Maybe here you can add another, the capacity to transfer the plasmid from one to another, transferring of this plasmid, what we call conjugation. The capsule, a virulence factor, then finally the lipopolysaccharide by the gram-negatives, and however also the ability to have a different nutrition modes can make them survive in the environment. So thank you for watching, and please subscribe and stay blessed.